everyone. In the previous lecture, we discussed about restriction endonucleases. We studied like what are the scientists who isolated restriction endonucleases. If you remember, restriction endonucleases discovery was contributed by three scientists, Arbor, Smith and Nathans, right? So we tell that restriction enzymes are widely used. They are widely used where in genetic engineering. And who are the scientists whom we can give the credit, like who first isolated restriction enzymes? In NCERT we have seen that in the year 1963 they started with restriction enzymes. They started in the year 1963. Two scientists, their name Arbor and Lin, right? Arbor and Lin, they identified restriction modification system in E. coli. This we have done it in previous class. They identified restriction modification system. So you can just look into the previous lecture to get this data. Then next, in the year 1970, we can tell that Smith, he isolated the first restriction enzyme and he called it as HIND2. In 1970, Smith isolated the first restriction enzyme and he named it as HIND2. So HIND2 identifies a 6 base pair sequence. What is that sequence called? GTC-GAC sequence. So he told it is a restriction enzyme, but its complete potency and all was established 5 years later. And the third scientist, here Arbor came, Smith came, and the third scientist is Nathans. This person used restriction enzymes for gene mapping and chromosomal mapping. So he observed restriction enzymes in E. coli and he told the system is restriction modification system, who Arbor and Lin. Then Smith isolated the first restriction enzyme and Nathan used restriction enzymes in gene mapping. For this discovery, these three people got Nobel Prize. Arbor, Smith and Nathans. These three people got Nobel Prize in medicine and physiology. They got Nobel Prize in the field of medicine and physiology. Children, in which year they got Nobel Prize? They got Nobel Prize in the year 1978. These three people got Nobel Prize in the year 1978. Whereas we studied Cohen and Boyer are also the backbones behind biotechnology, right? So they are the main people who gave the idea of RDNA technology. Then didn't they get Nobel Prize? They also got Nobel Prize, but it is in 1986, right? So the main pillars behind biotechnology are Cohen and Boyer, right? These people also got Nobel Prize. These two also got Nobel Prize in the field of, again, in the field of medicine and physiology only. But in the year 1986 they got. In the year 1986, Cohen and Boyer got Nobel Prize. Okay. So in our today's lecture is also going to be with restriction enzymes only, restriction endonucleases only. I'm just briefing, I'm just giving introduction who and all are the scientists who contributed towards restriction enzymes. Now, if you see what are restriction enzymes, they are the enzymes which cut the nucleic acids, right? So we can tell that restriction enzymes belong to a large class of enzymes called nucleases. So restriction enzymes, to which class do they belong? They belong to a large class of enzymes. What is that class called, children? Nucleases. Nucleases are the enzymes which break nucleic acids. Restriction enzymes are breaking nucleic acids. So a statement is there telling that restriction enzymes belong to a large class of enzymes called nucleases. Now we can categorize nucleases into two types depending upon how they are making the cuts. So we can classify restriction enzymes into exonucleases and endonucleases. We can divide the restriction enzymes into exonucleases and endonucleases. What are exonucleases? If they are cutting the DNA strands from the ends, 5' exonuclease will cut from the 5' end, 3' exonuclease will cut from the 3' end. Exonucleases, they cut the DNA from the ends. We can write exonucleases, they digest or they digest or they cut the DNA. From which end? They cut the DNA from the ends. Either from the 5' end or from the 3' end they cut the DNA. If we have to see the picture how an exonuclease acts, let us draw DNA randomly. So these are the bases. These are the bases: 5', 3', 3', 5'. And if an exonuclease acts, how it will cut the DNA? It will make the 5' end cut. For example, if you are thinking this much DNA it has digested, then the remaining DNA will be like this. So this is a 3' end and this again will be the 5' end and 3' end. So these bases are exposed because it has removed this much amount of the DNA and these are still intact. So exonucleases, what did I show? 5' exonuclease. Then I showed an exonuclease which digested the DNA from the 5' end. If you want to make it more clear, so we'll make a small fragment like this. These three got removed off and this is still intact with it. So such type of cuts we get when exonucleases act. Means like this type of cuts. If 5' exonuclease acts, from the 5' end DNA gets digested. If the 3' exonuclease acts, then from the 3' end DNA gets digested. So this is exonuclease. Coming to endonucleases. Endonucleases make internal cuts. They digest inside. So how do endonucleases cuts? Endonucleases cut inside the sequence. It cuts inside the sequence, right? So if we want to see how an endonuclease cuts, let us make a diagram and again see. So if there is a sequence like this, and if it is a restriction endonuclease, we are making blunt an enzyme. So it cuts in this manner, right? It cuts in this manner. Inside it is cutting. Inside is cutting. Then you will get a sequence in this manner. Blunt ends, sticky ends. These are sticky ends. So, right. Endonucleases, they are making internal cuts. It's not cutting at this end, nor it is cutting at this end. It is cutting in between. After 2 it is cutting. After 2 it is cutting. So when it cuts like this, so you will get sticky ends like this, cohesive ends like this. Now exonucleases cuts from the ends. Endonucleases cut inside. And again, if we take a plasmid molecule which is circular double-stranded DNA and I want to open the plasmid, then plasmid doesn't have any ends. Can the exonuclease cut a plasmid molecule? No. Then how can you cut a plasmid molecule? You need an enzyme which makes internal cuts. Then exonuclease can cut endonuclease can cut a circular DNA. Endonuclease only can cut circular DNA. Plasmid we need in biotechnology. Without plasmid we cannot work out because it acts as a vector DNA. Understood, children? What are exonucleases and endonucleases? Exonucleases cuts from the ends, whereas endonucleases make internal cuts. So means which enzyme are we using for biotechnology then? Can I tell that it is endonuclease we are using for biotechnology? Take a screenshot. Then we are going to talk about types of restriction endonucleases. Here we confirmed that we are going to use endonucleases in biotechnology. Now I am going to tell you what are type 1, type 2, type 3 restriction enzymes. Take a screenshot fast. Right. Hope you understood this. Now let us talk about the types of restriction endonucleases. There are 3 types of restriction endonucleases. We call them as type 1, type 2, type 3. If you remember molecular basis of inheritance, chapter 6, there we tell DNA polymerase 1, pol 2, pol 3. RNA pol 1, pol 2, pol 3. Like that, here we can tell there are 3 types of restriction endonucleases. So 3 types of restriction endonucleases are there. 3 types of restriction endonucleases are there. Type 1, type 2, type 3. Let us see the differences between them. The first one is called as type 1. Then the second one is going as type 2 restriction enzymes. And the third one is called as type 3 restriction enzymes. I will tell you that which type of restriction enzymes are important in biotechnology, which are commonly used are type 2. So, type 2, if we have to tell, so we can write down these are common type of restriction enzymes used in rDNA technology. So, this is a common type of enzyme which is used in rDNA technology. Then, why is it used? You will come to know. So, type 1, we'll start with type 1. Type 1 restriction enzymes are the simple or complex in structure. Means, it is made up of, the enzyme is made up of one subunit, made up of many subunits. It is a complex enzyme, it is made up of multi subunits. So, when we come to type 1, we will tell it is a complex enzyme. It is a complex enzyme made up of many subunits. It is made up of many subunits, we are telling. Now, coming to type 2, Amma, it is made up of one subunit only. Has one subunit only. Type 3 is intermediate, means it is not made up of one subunit like type 2, it is not made up of many subunit. We can tell it is of intermediate complexity, it is between 1 and 2, means it is not having many subunits, not having one subunit also. We can tell it is intermediate between, type 1 and 2. Intermediate between type 1 and 2 in complexity. Then, type 1 restriction enzyme identifies a recognition sequence. What is the length of the recognition sequence is it going to identify? It will identify 15 base pairs recognition sequence. Type 1 restriction enzyme identifies 15 base pairs recognition sequence. Whereas type 2 children, you know, in NCERT textbook they have given the recognition sequence for an enzyme called EcoR1. Its recognition site is palindromic, it is GAATTC. GAATTC 6. No, type 2 restriction enzymes recognition sites are of 4 to 6 base pairs only, not such big, right? So, let us write down here, write type 2 enzymes identifies 4 to 6 base pairs recognition sequences only. Good, no? Now, GAATTC is only difficult for us to remember 6 base pairs. How can we remember 15 base pair sequence? This is the recognition sequence. In this here it cuts also. You should remember. Difficult, no? That's why we are not using. Easy, no? That's why we are using. Coming to type 3, it has a recognition sequence of 8 base pairs. 6 to 8 base pairs recognition sequence it has. So, write down it identifies 6 to 8 base pairs recognition sequence. So, everywhere I think type 2 is proving it's better, right? Now, come on. Okay, it has identified, type 1 has identified this 15 base pairs recognition sequence. Now, after identifying it, it needs to cut it also, right? Nuclease means it has to cut the nucleic acid. Where it is cutting, we are waiting and seeing. Okay, it identified this sequence. Will it cut here? It came here, it just walked. It went out of this sequence. So, we are worried. It went out of the DNA where it is cutting. It will walk 1000 base pairs away from the recognition sequence. Thereafter 1000 base pairs it is cutting. Unnecessarily, it is again a burden to carry 1000 base pair sequence, right? So that means this way is also, it's not proving. The cleavage site for type 1 enzyme is 1000 base pairs away from the recognition site. I don't want it, right? So where is the cleavage site? The cleavage site is how many base pairs away? 1000 base pairs away from where? Away from the recognition site. It is 1000 base pairs away from the recognition site. Which one? Type 1 enzyme, it has to cut within the 15 base pairs, but it's not willing to cut here. It will move on 1000 base pairs, there it will cut. Unnecessarily this much you need to carry. That is the problem with type 1. Let's go to type 3 and ask where it will cut. It will cut 25 base pairs away from the recognition site, not 1000. It will cut 25 base pairs away from the recognition site. Let us write down here. Cleavage site is 25 base pairs away from the recognition site. That's for type 2. Type 3. Now come to type 2 and ask where are you going to cut? NCERT says that most of the enzymes, means type 2 restriction enzymes, which are widely used in biotechnology, rDNA technology, the cleavage site is within the recognition site. This is the recognition site. Here itself it is cutting. Not even one base pair unwanted it is carrying or it is loading. So that's why we use type 2 restriction enzymes. The cleavage site is within the recognition site. For type 2 enzymes we can tell the cleavage site is within the recognition sequence. It is within the recognition sequence. NCERT has given us the example. So there is an enzyme called EcoR1 GAATTC. This is a recognition sequence for an enzyme called EcoR1. This is a recognition sequence for an enzyme called EcoR1, and I am telling that the cleavage site is within the recognition sequence, means I need to show within this only. It cuts between G and A. It cuts this phosphodiester bond. It cuts this phosphodiester bond. So are we correct then? The cleavage site is within the recognition site? Yes. So that is why type 2 we are going to use widely in biotechnology, and this type 1 enzyme requires ATP for cleavage. It requires ATP for cutting the DNA. Whereas type 3 also requires ATP. This also requires ATP for cutting, for cleavage. Whereas type 2 doesn't require ATP, it just requires magnesium ion for cleavage. No ATP requirement. No ATP requirement, but it requires Mg2 plus ion for its activity. It requires Mg2 plus ion for its activity. ATP is not required. And if we have to list down some examples or names for type 1 enzymes, it is, one example is EcoK we can give, EcoP we can give. EcoK, yeah, EcoB children. EcoK and EcoB are examples of type 1. Type 2, how many are there? We wrote EcoR1. NCERT says that now we have 900 restriction enzymes which are available in the market. 900 enzymes are there available in the market, extracted from, they tell that extracted from 230 strains of bacteria. Extracted from 230 strains of bacteria. Restriction enzymes we cannot prepare on our own. Restriction enzymes are prepared only by the bacteria. From the bacteria we can isolate it. How many restriction enzymes have you isolated so far from the bacteria? Means 900 types of type 2 restriction enzymes you have isolated from the bacteria. From how many types of bacteria? 230 strains of bacteria. Coming to type 3 restriction, if you want examples, so EcoR1 we have already written, right? Hint 2 also comes here. HAE3 also comes here. PBU1, PBU2, PST1, clavin, SMAVin. Amma, don't think that one Roman letter you are using means it is type 1 enzyme, 2 Roman letter you are using means type 2 enzyme, 3 Roman letter you are using means type 3 enzyme. It's wrong. All these names what I wrote belong to type 2 enzymes only. This Roman letter denotes its order of isolation from that particular strain. Okay. And type 1 and type 3 we don't use it. Type 1 I have given two examples, Eco K and Eco B. For type 3 if we have to see, it is Eco P, Eco P15, Eco P, Eco P15 and Hind F3. These are the examples of type 3 restriction enzymes. Hope you understood what are nucleases. We have classified the nuclease into exonucleases and endonucleases. Exonuclease cut from the 5' end or from the 3' end. We don't want it because we want to make a cut at the internal side. Then we took endonucleases. Now we are telling that restriction endonucleases are of 3 types. 3 types of restriction endonucleases have been identified. Type 1, type 2, type 3. Type 1 is a complex in structure made up of many subunits. Type 2, it is simple in structure made up of only one subunit, and we commonly use this only in biotechnology. Type 3 is of intermediate complexity between type 1 and type 2. Type 1's recognition sequence is 15 base pairs long. Type 2 recognition sequence is 4 to 6 base pairs long only. Type 3 recognition sequence is 6 to 8 base pairs long only. In this way also, this is small. Then type 1, okay, it has identified 15 base pair sequence. Is it cutting within this 15 base pairs? It's telling no again. It's telling I'll walk 1000 base pairs. After that, I will cut. Then we don't want to take a load of 1000 base pairs. Its cleavage site is 1000 base pairs away from the recognition site. Whereas for type 2, the cleavage site is within the recognition site. Here, the cleavage site is 25 base pairs away, little away from this one. Now, this requires ATP for cleavage. 3 also requires ATP for cleavage, but this is not even asking energy also, but it requires magnesium ion. Then these are the examples, 900 enzymes which are there in the market, all of them they come under type 2 only. These are the examples. Quickly take a screenshot. Yes, can I rub this now? Now we are filtering, right? Exonucleases we filtered and we told we want endonucleases. Now we came to know type 1, type 2, type 3 are there. So we have chosen type 2 and we filtered type 1 and type 3 we don't want. Now again let us see these endonuclease enzymes identify recognition sequences. The recognition sequences are called palindromic sequences, right? So what we will tell, each restriction enzyme functions by inspecting the DNA sequence. Each restriction endonuclease functions by inspecting DNA sequence. It's moving on the DNA and identifying the sequence. Once it finds a particular sequence, it goes bind there and it starts cleaving the phosphodiester bond. We can tell that once it finds a specific sequence, once it finds a specific sequence, when the restriction endonuclease identifies a specific sequence, it goes and binds to that sequence. It binds to the sequence and breaks. What does it break? It breaks the sugar phosphate backbone, not the hydrogen bonds. It breaks the backbone present between sugar and phosphate. It identifies a sequence, it goes and binds to the sequence and breaks sugar phosphate backbone. Children, what linkage is there on the sugar phosphate backbone means? Phosphodiester bond. What bond is there on the sugar phosphate backbone? Phosphodiester bond means Restriction enzyme is cleaving which bond? Be very clear. It is not breaking the hydrogen bonds between the bases. It is breaking the backbone itself. Phosphorylester bond itself it is breaking. Understood? Now, when it is breaking, it is cleaving the DNA into fragments. Which type of ends do you get now? Basing on that, again we have two types. One type of DNA will give us sticky ends. Another type of DNA will give us blunt ends. After it digests, we will get two types of ends. One type of ends are called as sticky ends. There is another name for sticky end. Sticky end is also called as cohesive end. Sticky end is also called cohesive end. It has another name also. It is called staggered end. These are the synonyms, okay. Now, what are the other names of this end? They are called blunt ends. They are called even ends. They are also called flush ends. What are the other types of ends which are produced called as? They are called, you will understand why is it called blunt end when I draw it. They are called blunt ends. They are called flush ends. Or you can call them as even ends. That means there are some enzymes, when they cut the DNA, they give blunt ends. There are also some other restriction enzymes. When you cut them, it will give sticky ends. So, we will see the examples. We will see how it cuts. Then you will get a better clarity. For example, we know one enzyme, EcoR1, which is very much given in NCERT, which gives a sticky end. We will go with that. Okay. So, I am taking an enzyme. The name of the enzyme is EcoR1. Is the name of a restriction enzyme. The first alphabet states its genus. The next two alphabets tells its species. The fourth alphabet tells its strain. One is the order of isolation. EcoR1 is isolated from a bacteria called Escherichia coli. Escherichia coli. This enzyme is isolated from this bacteria, and what is its recognition site? We will write. And what is its cleavage site also we will write. Its recognition site is GAATTC. GAATTC, 6 base pair sequence. Opposite end is GAATTC. Such type of recognition sequences identified by restriction enzymes are called palindromes. What do you mean by palindrome? In English, we tell madam is a palindrome. Madam this way also, madam that way also. In NCERT, they have given Malayalam as a palindrome. Malayalam this way also, Malayalam this way also. English is only one sentence, one way you can do it. But in DNA, which is 2 strands, we call palindromic sequence with rotational symmetry. Palindromic sequence with rotational symmetry. 5' to 3', whatever sequence is there, 5' to 3' sequence you have in rotation. 5 to 3, GAA, GAA, TTC, TTC. Such sequences are called palindromic sequences. It has identified. This is the recognition sequence. Now, where it will cut? It will cut between G and A. Here also, G and A, here also. Means this phosphodiester bond, it is breaking. This phosphodiester bond, it is breaking. So, means we discussed that it's a type 2 enzyme and it cuts slightly away from the center, but within the recognition sequence. So, see, within the recognition sequence, it is cutting. Yeah, very much within the recognition sequence, it's cutting. But not at the center, children. It's cutting slightly away from the center. Now, when it cuts like this, how the fragments are generated? The fragment will be like this. G will be here. C will be here. Because it has digested here, T, T, A. Overhanging stretch will be there. This is 3' means this is 5'. Opposite amma, A, A, T, T, C and G are together. This is 5, this is 3. If this is 3, this is 5. 5 prime overhanging stretches you are getting. Is it a sticky end? Yeah, it is sticky. It is sticky because TTAA can pair with AATT. They are complementary. They can make hydrogen bonds easily. Ligase can aram se join it to give a rDNA molecule. So now that means echo R1 gives sticky ends then. So echo R1 is an enzyme extracted from Escherichia coli. Its recognition sequence is GAA, TTC. Its cleavage site is between G and A. After it cleaves, the DNA fragments are like this, single-stranded, overhanging stretches. 5 prime overhanging stretch you will get, which are complementary, which can easily make hydrogen bonds among themselves. So one enzyme I showed you. Shall we see some more examples also? These examples will get repeated for us when we study PBR 322. In that order only I will go you. Now it will be difficult for you, but it will help you when you are studying PBR. Let me go with the second enzyme, which also gives sticky end, is called as clavin. The enzyme name is clavin. Is extracted from caryophenolatum. It's extracted from caryophenolatum. Now, the recognition sequence of clavin is ATC. What is it? It is 5 prime. I'll tell you one more magic here. Now, it is a 6 base pair sequence. Let us not remember all the 6 base pair sequence which will be loaded for us. We'll take only 3 base pair sequence. The other 3 will follow because it's a palindrome. I'll tell you how it comes. For clavin, you just remember ATC. Clavin ATC. See, this is A na. Make this L as T. This is C. ATC. ATC is the sequence. Like that you remember. Now, the other 3 will follow. I will tell you how does they follow. So, since it is palindrome, in opposite, it will be A, T, C, right? Now, this 3 you can fill based on the complementarity. C pairs with G, T pairs with A pairs with T, C pairs with G, T pairs with A pairs with T. Means ATC, GAT is a palindromic sequence for clavin. And where does it cuts? It cuts between after AT here, then that's a cleavage site. How does it generate the fragments also? Let us see. It generates the fragments when it cuts like this. A, T will be together. This 4 I need to write. A, T, A, G, C only. This much only. Opposite strand will be CG, AT, TA, 5 prime, 3 prime, 3 prime, 5 prime, right? So this is 5, then this will be 3, this will be 5. So this is the sticky end what clavin has given. So now it will be difficult, but if on practice it will become clear for you. For echo R1 we are familiar, it is GAA. Only 3 we can remember now. TTC will follow as the way I told you. GAA is for echo R1. For clavin I told A, make this as T, C, ATC. It cuts after AT. Now shall we go with the third enzyme? The third enzyme is HIND3. The third enzyme is HIND3, which is isolated from a bacteria called Haemophilus influenzae RD strain, which is isolated from bacteria called, we'll write with blue only, Haemophilus influenzae. Haemophilus influenzae RD strain it is. Now, what is the sequence? For HIND3 means it is AAG. Amma, for echo R1 it is GAA, no? Come ulta, AAG. Write down it here. So it is AAG. I asked you to just remember 3, the other 3 will follow. So we need to fill it. We will do that. Come this way and tell A, G again. A, G. Now G pairs with C, T, T. G pairs with C, T, T. So this is the sequence for Hind3. Hind3 is also an enzyme which is going to give us sticky ends only, cohesive ends only, which is isolated from bacteria called Haemophilus influenzae RD strain, and its recognition sequence is AAG CTT. And where does it cut? Between the first A and the second A. It makes a cut here, children. Now we will see how we get the fragments then. So it is an uneven, it's going to give sticky end again. So let's do that. So only A will be there. T, T, C, G, A also. And this end, it will be A, G, C, T, T and A. Now see again sticky end. So far how many we have seen? 3 with very easy. Eco R1, GAA, clavin, ATC, Hind3, AAG. Fourth enzyme is BAMH1, which is isolated from a bacteria called Bacillus amyloliquefaciens HD strain. Write down. B for Bacillus, am for amyloliquefaciens. It is HD strain. And BAMH1 recognition sequence is GGA. 2 times G now. So here you gave 2 times A. Now give 2 times G. It becomes GGA. So it will be 5 prime GGA. We will do that. Okay. GGA. A pairs with T, G pairs with C, G pairs with C. T, C, C. This is a palindromic sequence identified by BAMH1, and the cleavage is between GG. The cleavage site is between first and second. So here also between first and second, here after 2, again first and second. Now here also I am telling it is between first and second. Then what fragments you get? Again it's an uneven cut only, means it's going to give sticky end only. So it will be G, here it will be C, C, T, A, G, and this side it will be G, A, T, C, C, G. Now see, C, T, A, G can pair with A, T, G, C. This is what is called sticky complementarity. Let's take one more enzyme, SAL1. From Streptomyces albus. SAL1 restriction enzyme is isolated from bacteria called Streptomyces albus. So fourth alphabet is not there, and its recognition sequence is GTC, GAC. If you remember, it is for Hind2. So the same sequence, SAL1, Hind2, same sequence. So it is GTC, GAC. Hind2 is the first enzyme identified by Smith which makes sticky end. For the same sequence, this is giving blunt end. Okay. So GTC, GAC. It cuts here with black. I am showing the cleavage sites. No, it cuts here. It cuts here. Now the fragments generated will be G. It is cutting, right? C, A, G, C, T will be here, and here it will be TCG, TCG, A, G and G. Again complementarity. Okay. So how many we have seen so far in the board? Eco R1, GAA we know. Clavin, CTC. Hind3, it is AAG. BAMH1, 2 Gs, 1A. SAL1, GTC, GAC we remembered for Hind2, right? The first restriction enzyme, you have to remember it. So SAL1 also copied the same thing. What Hind2 is doing, SAL1 also copied the same. Then 2 more enzymes are there. What are they? PVU1. Fifth one is PVU1 from Proteus vulgaris, from Proteus vulgaris, okay, and its recognition sequence will be CGA. What it is? CGA. Opposite will be T. A pairs with T, G pairs with C, C pairs with G. So CGA, TCG. This is the recognition sequence for PVU1. Cleavage site not after 1, not after 2, after 4. It is jumping more. Here it is after 1, here it is after 2. But PVU after 4. Here between T and C. Between T and C. I am not writing that ends. Okay. Then the last enzyme is PST1. So what am I taking means all these enzymes will repeat for us when we see PBr322 vector. In PBr322 vector, all the restriction enzymes which are there except PVU2, all are sticky ends only. Hint 3, BAMH1, SAL1, PVU1, PST1. PST1 is isolated from bacteria called Providentia stroti. Providentia stroti. And the site for PST1, shall I write here? It will be CTC. It will be CTC. It will be CTG, CAG, CTG, CAG. So I wrote for PST, I wrote here. And its cleavage site, this is cutting after 1, this is cutting after 2, this is cutting after 4, this will cut after 5. 1, 2, 3, 4, 5. It cuts here. It cuts here. So take a screenshot. All these 7 enzymes are making sticky ends or cohesive ends or staggered ends. Right? Took the screenshot. So what are we discussing? We are talking about endonucleases. We want type 2 restriction endonucleases. Type 2 restriction endonucleases, they inspect the DNA, they identify a sequence, they bind to the sequences and they break which bond on the DNA, phosphodigester backbone they make. When they are breaking, they give 2 types of cuts. Sticky cuts, which are also called cohesive ends, which are also called staggered ends. We are seeing, yeah, it is sticky, again sticky, again sticky, again sticky, again sticky. I don't have space here. I didn't draw for this one, but I showed you the cleavage sites. You can draw it, I think. Then all these are sticky and enzymes only we want in biotechnology. You took a screenshot. Now I am going to rub it and we are going to see similar examples for blunt ends also. We are going to see similar examples for blunt ends also. Can I erase this now? And I am telling that we need sticky and enzymes only. We need sticky and enzymes only. But we have to look into what are the enzymes which can make blunt enzymes also, no? Because sometimes in MCQs, they will give and they'll ask you to identify odd one out. 3 enzymes will be sticky and digesting, 1 will be blunt end. So when you know who are sticky and who are blunt, then only you can eliminate them, no? So if you want, shall we list down the examples here? For sticky ends, what is our list? Can I write here? EcoR1 is our first enzyme. After EcoR1, I end with clavin. After that, Hind3. After that, BAMH1. After that, SAL1. After that, we went with PVU1, PST1. Amma, all these are examples for sticky ends. Remember them. So now blunt end names I will give. So now if they give anyone and ask you to identify, from this you can eliminate. The examples for blunt ends are, blunt ends, I'll start from here. The first enzyme is ALU1. The first blunt end enzyme is ALU1, which is extracted from a bacteria called Arthobacter luteus. ALU1 is extracted from a bacteria called Arthobacter luteus. Now, what is its recognition site? What is its cleavage site? Recognition site is 4 base pair sequence. It is AG. Other two will follow, no? We'll write down. From here, AG, G pairs with C, A pairs with T. G pairs with C, A pairs with T. Amma, this is a blunt-end enzyme. Blunt-end enzyme means it has to cut at the center. It has to cut at the center. Means the cleavage site is here, children. Then you will get a DNA strand like this. This side AG, this side CT. This side AG, this side CT. Did you see the blunt end? It is blunt. Sticky ends are not there. Single stranded overhanging stretches are not there. Do you want such type of DNA fragments? In such type of DNA fragments, if we try to insert our desired DNA here, like this, if you try to insert your desired DNA, if you just apply pressure here, your DNA will be out. Stability is lost. Experiment gone to ruin. So that is why we don't want blunt end enzymes. We want sticky ends. So ALU 1, it gives such a type of cut. Let's take a second example. Hind 2 we already know. Hind 2 is the first enzyme isolated by Smith from Haemophilus influenzae. GTC, GAC, we already did it. The sequence is GTC, GAC, GTC, GAC. Repeat with me so that you will become familiar. Now, blunt end, no? Center, no? Let's do it. Here and here. Then the strands will be, yeah, GTC, GAC, GTC, GAC. Again, blunt ends only we got, means Hind 2, the first enzyme what Smith isolated is not a useful enzyme. Then the third enzyme is HAE3. What is the third enzyme? HAE3, which is isolated from Haemophilus aegyptus. It is isolated from Haemophilus AEGYPTUS. Haemophilus aegyptus. Recognition sequence is 4 base pair sequence GGCC. Easy. GGCC. GGCC. Blunt end, no? Center cut, no? Do that. This phosphorus bond, this phosphorus bond. Then we get blunt ends. GGCC. CC, GG. Don't want again. One more example we will do and we can end the class. So, ALU 1, blunt end enzyme. Hind 2, blunt end enzyme. HAE3, blunt end enzyme. The last blunt end enzyme which is there in PBR vector is PVU2. What is it? It is PVU2 from Proteus vulgaris. Its recognition sequence is CAG, CAG, CTG, CAG, CTG. Blunt end enzyme, no? Center cut, right? Here and here. Then your DNA fragments will be blunt. Flush ends, even ends, CAG, GTC, CTG, CAG, CTG. Yeah, CTG, right? So, I completed. So, we completed everything related restriction enzymes by taking 2 lectures. In one lecture, we have seen the nomenclature, we have seen who isolated it and what is restriction modification system, what are palindromes. In the previous lecture, we discussed. Now, in this lecture, we discussed what are exonucleases, what are endonucleases. We want endonucleases. Okay, right. under endonucleases, type 1 are there, type 2 are there, type 3 are there. We want type 2. What are the advantages? Clearly we have seen that. Then type 2 can give two types of ends, sticky ends, blunt ends. Do you want this? No. We want sticky ends. Sticky end enzymes, 7 enzymes I have listed. Blunt end enzymes, 4 enzymes I have listed. Now if they ask you a question, identify enzyme which can give blunt end. Yeah, you know the 4. Identify an enzyme which gives sticky end. Check for these options, check for these examples in the options. Or I identify the odd one out. 3 enzymes will be blunt with 1 enzyme which is sticky. This is odd one out. 3 enzymes will be sticky, 1 will be blunt. This is odd one out. So go through that carefully so that you can work out on restriction enzymes very well. Now in the next lecture, we're going to talk about separation and isolation of the DNA fragments. Hope the lectures are informative. If you like the content, like, share and subscribe to my channel. Thank you.